Well, good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled Pushing the Limits of PCR, Considerations for Assay Development in High-throughput qPCR and Digital PCR Platforms. My name is Andrew Jordan, and I'll be your Xtalks host for today. And a big thanks to LGC Biosearch Technologies who helped develop the content for this webinar. LGC Biosearch Technologies is a global leader in the design, development, and manufacture of custom oligonucleotides and associated reagents for the molecular diagnostics, research, and applied markets. LGC Biosearch provides best-in-class products for use in quantitative and endpoint PCR, utilizing its extensive portfolio of proprietary fluorophores and dark quenchers. In addition, LGC Biosearch has expertise in IP related to qPCR probe design and RNA fish. And now I'd like to introduce our speakers for today's webinar. Luke Linz is the Laboratory Operations Manager at LGC Genomics in Alexandria, Minnesota. He is responsible for overall operation and administration of the laboratory with specific focus given to customer needs, research requirements, assay development, quality control, and workflow processes. Now, Linz joined LGC Genomics in 2013 after receiving his PhD from Iowa State University, where he conducted research in the field of microbiology. And your second speaker for today, Jim Huggett, is a principal scientist at LGC and a senior lecturer at the University of Surrey. After receiving his PhD at Cardiff University, he then spent 7 years as a senior research fellow at University College London, investigating molecular diagnosis of infectious diseases in a developing world setting. Jim's current interests include genetic and genomic analysis applicable to infectious diseases and other fields such as cancer diagnostics. And with that, I would like to hand the presentation control and microphone over to your first speaker for today, and that's Dr. Luke Linz. Dr. Linz, you can begin your presentation when ready.
All right, Andrew, thank you very much for the introduction, and welcome to everyone joining us today. The title of my presentation is Robust Assay Development for Multiplex qPCR and SNP Genotyping in Miniaturized Volumes on a High-Throughput Instrument. I'd like to start with an outline of the presentation. I've broken it into two parts for today's talk. The first part is going to highlight the use of BHQ Plus probe assays and the IntelliCube real-time PCR instrument for multiplex SNP analysis, and we will highlight examples of data from various research areas. The second part of the talk, I'll switch gears and get into gene expression and the use of BHQ probe for qPCR assay design, and talk about multiplexing considerations and highlight some of the data we've been able to generate. I'd like to start with just a definition of the SNP. A SNP, a single nucleotide polymorphism, is a form of genetic variation where you have a single nucleotide change between individuals or between a pair of chromosomes that would otherwise be identical. And an allele is, by definition, an alternative form of a gene or SNP, and we'll hear this term used frequently throughout the presentation. SNPs are important in a variety of research applications, including human research, where they can serve as biomarkers for disease prediction and are also important in predicting response to certain pharmaceutical drugs. SNPs are also important in the ag biotech sector, where they are used in plant breeding programs for marker-assisted selection or backcrossing, and are also important in trait associations and can be used to track certain animals or plants in these research programs. I will now get into introducing the BHQ Plus chemistry from LGC Biosearch Technologies. BHQ Plus probes have a duplex stabilizing technology that allows them to form stable duplexes with their DNA targets. This leads to enhanced target specificity, which is very critical when looking to discriminate between different SNPs. They are dual labeled, so they have a fluorophore and black hole quencher, and this black hole quencher does not interfere with fluorescent dye signal after hydrolysis. Another nice feature is the real-time design software offered on the Biosearch Technologies website. This allows users to input their target sequences and get assistance in designing BHQ plus assays. So a typical probe-based SNP assay consists of a forward and reverse primer and 2 allele-specific probes. These probes are designed to discriminate between the SNP of interest. In this example, we are looking at a C to A SNP. So you would have one probe labeled with a fluorophore specific to the C allele and another specific to the A allele. And this probe would have a different fluorophore. During the PCR process, you get annealing of your primers and allele-specific probes to their intended targets. And through primer extension, the probe is then hydrolyzed by the exonuclease activity of the Taq polymerase. This causes the release of the fluorophore from the quencher and gives you that fluorescent signal that can be detected by your PCR instrument. So using fluorescence-based assays, we can monitor our reactions in real time, but for SNP analysis, it's really that endpoint signal that we are interested in, and that is plotted in a cluster plot. In our example here, we are showing the cal-fluor orange 560 signal plotted in the Y direction, and the FAM signal for our assay plotted in the X direction. So we can use these plots to score our data. And so in the example here, I've highlighted the blue clusters. So these would be samples or individuals that had generated only cal-fluor orange signal, and therefore they are homozygous for the A allele. The middle cluster, or the purple dots, generated FAM and cal-fluor orange signal, and therefore are heterozygous, so they contain both the A and C allele. And finally, the ones that are the red dots here have only the FAM signal and are homozygous for the C allele. So now I'd like to introduce the platform for which this work was done. And this is the IntelliCube real-time PCR instrument. This system is integrated liquid handling, amplification, and detection all in one unit, and is the latest development of the array tape automation. For those of you who aren't familiar with array tape, it is a thin, flexible polypropylene consumable consisting of embossed reaction wells that have a working volume of 1.6 microliters. Now this array tape serves as a replacement for your standard microplates or PCR plates. The tape we are showing here in the bottom right comes as a unique 768 well format. Now, the IntelliCube system has a 5-channel fluorescence detection. The IntelliCube can be hooked up to your laboratory's network and can therefore be accessed by a PC in order to generate your protocols. DNA sample plates are prepared and loaded onto the system and has a 10-plate capacity for sample plates. This can be DNA or RNA samples. It has 3 stations for loading your assay plates, which would contain your master mix and your primers and probes, a touchscreen interface to start your protocols, and after the protocols have run, you can then analyze the results again on your PC. So just breaking that down a little bit more, the first step after starting your protocol, the IntelliCube will dispense your samples. This is an 800 nanoliter dispense, and then your assay, also 800 nanoliters to create that 1.6 microliter reaction. The system automatically seals the array, moves it into the thermocycling station where it will amplify your DNA through the PCR process and collect data in real time. Now this is a continuous process, so as soon as one array is finished, moves out of the machine, the next array is dispensed and ready to go, moves into the thermocycling station, and continues the PCR process. The IntelliCube optics fit well with the proprietary dyes from Biosearch Technologies. These are the cal-fluor and quasar dyes. We have a CCD camera, uses filtered LEDs, and can capture all 5 channels in 15 seconds or less. So when we talk about multiplexing for SNP analysis, we can break that down, and the dyes we use I've broken down in the table at the bottom. For the first SNP, allele 1, we can use FAM, allele 2, calfluor orange 560, and our second SNP, in the same well, we can use calfluor red 610, quasar 670, and we use a passive reference, in this case, as TAMRA. And what I'm showing here is the emission spectrum of those dyes, and you can see how those are separated out. So over the next several slides, I'm going to highlight data that we've generated in various research areas and show you the multiplexing capabilities of the chemistry and instrument combination. So for our human SNP genotyping study, we use purified genomic DNA from blood samples, diluted these to 5 nanograms per microliter, and designed assays using that real-time design software. So these are BHQ plus SNP assays. And for this study, we used a clear call master mix from LGC. We used 4 different SNP assays. These are known to be important for drug metabolism, so variation in these SNPs can impact an individual's ability to metabolize certain drugs. The first SNP is VCORC1, and then there's 3 enzymes from the cytochrome P450 family. So the first slide here showing some data is in singleplex. So all these assays were run 1 assay per well, and we're showing the cluster plots for the 4 assays. So what we're looking at here again is calfluor orange signal plotted in the Y direction and FAM signal plotted in the X direction. And we can look at our cluster plots. And what we're looking for here is, do we have good separation of our clusters and good separation as far as signal to noise. So from our NTCs, our no template controls, which are indicated by the black dots in the lower left corner, are we getting sufficient separation? And you can see here we have good clusters, well separated. And what you might notice for this is we only have 2 clusters instead of 3. For a lot of these SNPs, there are rare alleles, so we didn't have any in our population here, any homozygous mutant samples. The next slide is showing the same assays but in a duplex format. So this is running 2 SNP assays in a single well. And so I've broken it down by duplex 1, duplex 2. So for duplex 1, we're running the C9 star 2 and VCOR assays. So star 2 was run with the same probes as I showed previously, the calfluor orange and FAM, and the VCORC1 in this case was used Q670 and calfluor red 610. And what we can see is we were able to maintain very good cluster quality, separation of our clusters in order to make them easily scorable, and we maintained a very good signal to noise ratio. Duplex 2, we saw a very similar thing, easily scorable clusters. So just to reiterate, this method is utilizing all 5 channels of our detector, 4 channels for our 4 allele-specific probes, and a reference dye as the 5th channel. The next area where we wanted to demonstrate this capability was for bovine SNP genotyping. For this, we had acquired 29 beef samples from a variety of different grocery stores. Instead of using purified DNA, we used a sodium hydroxide method for a crude prep. And so basically this involves lysing the cells in a sodium hydroxide solution, followed by neutralization in a TRIFS buffer. We then designed assays, again using the real-time software, and used a clear call master mix for the testing. These SNPs, we dug into the literature and found some various SNPs that were important for commercially relevant traits such as meat tenderness and fat content. The SNPs are listed here with their genomic information. First was leptin, a calpain gene, a calpistatin gene, and a promelanin concentrating hormone. So following the same format from the human data, we ran all of these assays in singleplex. In this case, we did have 3 clusters for all of our assays, but we still maintained very good cluster separation and good signal-to-noise ratio. So all of these in singleplex were just with CalFluor Orange and FAM. Flip to the next slide, and we're looking at the duplex version of these assays, where you can see we maintained very scoreable clusters, good separation, and just to reiterate, the same with the human DNA, we were achieving the exact same genotype calls regardless of the format in which we tested them. The last example, we wanted to show this in plants. So this is with corn or Zea mays. Used heirloom corn seeds, and again, a sodium hydroxide prep method to get a very crude extraction of these samples, BHQ plus assays, and clear call master mix. To identify these SNPs, we again looked in the literature and found a publication where they had identified highly polymorphic SNPs, and we picked these not to be associated with any specific traits of interest, purely just because of their polymorphic nature, and we wanted to use them to demonstrate instrument and assay performance. So showing the same kind of format here, these are the 3 assays in singleplex. So this is very crude DNA, BHQ plus assays, and the clear call master mix. We see good cluster separation and a very good signal. I'm going to flip to the next slide, where we show these again in duplex, 843 and 115 duplex together, 176 and 611 duplex together, using all of the same dyes we used before. So our SNP1, CFO and FAM, and SNP2 within a duplex, our Quasar 670 and CalRed 610. And we were able to maintain our genotype calls between singleplex and duplex, and also very good cluster separation and signal to noise. So with that, I'd like to summarize the first part of the talk. What we were hoping to demonstrate here was successful duplex SNP analysis using the IntelliCube real-time system with BHQ plus assays in array tape. And just to reiterate, these are 1.6 microliter reactions. And we were able to maintain our data quality as documented by good signal to noise ratio, cluster separation, and maintaining the calls from singleplex to duplex. And so effectively what this does is allow us to double the number of data points that is achievable with each run. So in our 768 well array, we can really get 1,536 answers from a single array. And we've shown that clear call and BHQ plus probes are an effective combination in a variety of research areas with different sample inputs. So whether that's purified DNA or very crude samples, we are able to see very good data from both methodologies. So I'm going to switch gears a little bit and now get into gene expression using BHQ probes. Just a quick summary of gene expression. It's the process by which genes are transcribed to synthesize functional gene products. Gene expression is important in a variety of applications, including cancer research, drug screening and development, and in agrogenomics. Just the diagram on the right is just showing a very generalized description of how our genomic DNA is transcribed into RNA or a messenger RNA template that is then translated into a functional gene product. There are several methods to analyze gene expression. These include microarrays, RNA fish probes, next generation sequencing through RNA-seq. But what I'm going to focus on for the remainder of this talk is reverse transcription quantitative PCR, or RT-qPCR. So again, just the process of RT-qPCR, we are looking at that messenger RNA, and it first undergoes a reverse transcription step to create the complementary DNA strand, or cDNA, which then undergoes a standard PCR process to get a final PCR product. There are a few different ways we can do this, through either a one-step RT-PCR or two-step RT-PCR, and there are advantages and pros and cons to each of these methods. A one-step process, the cDNA synthesis and PCR occur in the same reaction, so this reduces the pipetting steps, reduces the number of reactions that you need in order to get your answer. However, with a two-step process, the reverse transcription occurs in a separate reaction, so this gives you a little bit more flexibility. If you're looking for many targets from a single RNA sample, you can create that cDNA from that sample. It's more stable, and you can screen many targets against that single sample. So to demonstrate some assay development and multiplexing capabilities of our IntelliCube system with BHQ probes, we wanted to find some relevant targets. So we looked through the data, identified targets that were differentially expressed in various tissues. So we have 3 genes of interest that we'll be looking at today: mucin-1, or MUC1, hepcidin antimicrobial peptide, or HAMP, and a solute carrier family 34 member 1 protein, or SLC34A1. And we chose some common reference genes: a Tata box binding protein, TBP, glycerol-aldehyde 3-phosphate dehydrogenase, or GAPDH. However, I must note here that there are no universal reference genes, and so the stability of multiple genes should be evaluated for each individual study, and we can't assume because a reference gene is a common reference gene that it's going to work for your particular study. But for our purposes of demonstrating assay design and PCR performance, we chose 2 to work with. And in the absence of having a large pool of samples to choose from, we purchased commercially available human RNA from multiple sources, from multiple source tissues, and designed BHQ assays with the real-time design software. And we decided to use a one-step RT-PCR workflow on the IntelliCube, although both a one-step and two-step process would both work. So I just wanted to walk through some of the assay design steps. First, identified our sequence information. So we're looking at transcript sequences and wanted to design cDNA-specific assays and avoid the amplification of genomic DNA if possible. So we did this through designing exon-spanning assays to hopefully avoid identification or detection of genomic DNA in those samples. The BHQ probes and primers were designed with the real-time design software, and one of the approaches you can take to speed up the design process or the validation process is to just get multiple designs for each assay and test them out initially. This saves time and having to go back and redesign if necessary. Primer considerations. Primers are one of the more important things. We need to understand if our primers that we're going to be used, especially when we're multiplexing, are compatible with each other. So are we forming primer dimers with other assays or within an assay? And there are several software programs available to assist with this. Also, looking at any off-target interactions. So when we're designing these assays, are they specific to our target, or are we going to be amplifying targets that were not intended? And there's several blast programs you can use to use a primer blast to understand what targets am I amplifying. And during the assay optimization process, we always start with understanding what the performance of the assay in singleplex, and then adding assays sequentially when we start multiplexing. So we like to start, after we've designed and acquired our primers and probes, start by characterizing the assay using a synthetic DNA template. So what we're looking at when we're looking for qPCR performance is specificity, sensitivity, and PCR efficiency. And when we're looking at gene expression studies, we ideally want to have very similar efficiencies across all of our assays. So we took our synthetic DNA, in this case, and did a dilution series from 10 to the 7 down to less than a copy per reaction, and used 8 replicates per dilution. The more replicates you have, you get a more precise or more accurate answer when it comes to your efficiencies and your limits of detection and so on. For our assays, we use a standard 200 nanomolar probe concentration, 600 nanomolar primer concentration. These can vary depending on the assay or the instrument you're using, but there are standard ranges which are appropriate. So when assessing assay performance, I just wanted to define some of the criteria we're using. Limit of detection, so what is the lowest amount of analyte in a sample that can be detected but perhaps not quantified. Usually this is associated with a stated probability. So assuming a 95% confidence interval, actually the theoretical limit of PCR is 3 molecules. Whereas the limit of quantification, so what concentration can be detected while meeting the defined requirements for error and therefore can be quantified. So a standard method is to use a 35% coefficient of variation. So we're looking at across our dilution series in our standard curve, at what concentration are we exceeding that variation, that 35% CV mark. And then it's the concentration prior to that becomes your limit of quantification. R squared, so how well is our a trend line fitting to the data? So our standard curve, a value of 1, obviously being a perfect fit. And PCR efficiency, which we're calculating from the following formula that's shown. And this is related to the slope of the standard curve, a slope of negative 3.32 equating to 100% efficiency. So this is an example. Before I show all the data, I'm going to start with just gap DH and look at assay performance with our synthetic target. So at this point, it's not RT-PCR. We're just looking at how well our primers and probes working, and we're looking at a singleplex format of this assay. And so in our dilution series, we actually broke it down into 10-fold dilutions down to 1,000 copies, and from there did 2-fold dilutions in order to get a more accurate LOD and LOQ, or limit of detection and limit of quantification. So we can look at our amplification curves on the left, and we plotted our standard curve with quantity along the x-axis and CQ value in the y direction. So now looking at all 5 assays, the 3 genes of interest, and the 2 reference genes, we can see very good amplification curves, and the data is actually reflected in the table in the lower right. So we're looking at our LOD, LOQ, R squared, and PCR efficiency. So we can see we had a very good limit of detection, 3 to 5 molecules, and these are estimated copies based on the synthesized synthetic DNA. Remember, with our 95% confidence limit, the theoretical limit of detection was 3 molecules, so we're right down at that theoretical limit. Our limit of quantification was also very good, 15 to 30 copies. R squareds approaching 1, and percent efficiency 96% or above. And typically with PCR assays, generally 90 to above 90% is typically accepted. But again, we want, when we're doing a gene expression assay or study, we want to have all of our assays very similar in PCR efficiency. And just to note here, our LOD and LOQ calculations were performed using a GenX software from MultiD. And the values we're showing for LOD and LOQ are in target copies, not necessarily a concentration, but actual target molecules. So we next all these assays would perform using an one-step RT-PCR process because it's important to evaluate the performance of your assay in the sample matrix expected for the experiment. So we created a standard curve from 50 nanograms down to 5 femtograms. And on the next slide here, I'm showing the assay performance with the purified DNA in a singleplex format. In this situation, since we have varying levels of gene expression, we weren't looking for LOD or LOQ, but looking at R squared and percent efficiency. And I noted that a few of our assays, since they were lower expressing, we only had 3 points on our standard curve, which is not ideal. Ideally, we like to have more than that, but since due to the low expression, we weren't able to achieve that. So the take home here was that we were able to maintain very high PCR efficiencies switching to the purified RNA sample. So now, getting into the multiplexing considerations. The first thing is we want to make sure we have a master mix that's capable of multiplexing. Not all master mixes are created equal. They need to be formulated with adequate amounts of enzymes, DNTPs, magnesium, etc. It makes sense that we're amplifying more targets. We need to have a sufficient amount of these components in the mix. Primer concentration is very critical as part of the optimization process. Typically, it's a reduction in primer concentration that's going to be necessary, and it's possible too if you have a very highly expressed target. We can limit the primer concentration of that target, and so that's not using up a lot of the materials in your reaction. And like we mentioned before, we start in singleplex and we add assays sequentially in order to see how performance is impacted. And our goal here when multiplexing is to maintain performance as far as sensitivity, efficiency, and similar CQ values. So when I start multiplexing, I want to make sure that I'm getting the same CQ values if I'm running the exact same samples. So for our multiplex example, we chose to use 3 targets and a passive reference dye. So we were utilizing 4 of the 5 channels on our IntelliCube. Target 1 was FAM, target 2, Cal4 Orange 560, target 3 was Quasar 670, and our passive reference was ROX. So this is an example of how why primer optimization is important. What I'm showing here is an example from our TBP assay. So we're showing amplification curves from a triplex assay, but I'm only showing the TBP curves from that assay. On the left, you'll see when we ran it with a very high primer concentration of the other 2 assays, the MUC1 and the GAP-DH. We saw at our lower concentrations we had a fanning effect. The curves didn't look good, and we were getting a shift in our CQ values because of it. Now, when we lowered our primer concentrations and empirically determined what was working best, we saw that when we did lower those primer concentrations, we were able to fix those curves. We were seeing better CQ values that were in alignment with what we were hoping to see, and just an overall improvement in signal. The next slide shows the triplex assay performance. So we started with 5 assays, and we wanted to focus in on 3 assays for the purposes of demonstrating multiplexing. So what we're looking at here is singleplex, duplex, and triplex for our 3 different assays, and we can see that the real-time curves maintain their shape. It's going to be hard to really focus in on the performance until I go to the next slide, but just visually we could see we were getting very good PCR performance in a singleplex, duplex, and triplex format. So the table here breaks that down. We're looking at our, again, LOD, LOQ, and percent efficiency in singleplex, duplex, and triplex with our 3 assays down the left-hand side. So you can see we maintained very good specificity. We did see a slight increase in our limit of detection when triplexing, but there is always something we have to give up when multiplexing, although we were able to maintain our limit of quantification across the board regardless of the plex. And PCR efficiency was maintained singleplex, duplex, and triplex. And what we're showing in the bottom half of the slide is just an overlay of the curves from singleplex, duplex, and triplex of our GAP-DH assay. And what we're hoping to show here is that we didn't see a significant change in CQ value, regardless of whether we were looking for one target or 3 targets in that well. So after we had characterized our triplex assay, we wanted to see if it would maintain performance, or we would actually get the same gene expression data using that triplex assay versus if we ran all of those assays in a singleplex format. So we decided to look at MUC1 gene expression in kidney, liver, and lung tissues with the goal of achieving comparable results using a singleplex and multiplex format. We repeated this experiment 3 times in order to assess run-to-run variability of our instrument and the chemistry, and used a comparative CQ approach for relative quantification. This slide shows the data. So we ran 50 nanograms of RNA per reaction, used a one-step RT-PCR approach, and we're showing the curves in singleplex, triplex, and then overlay of the 2 formats for our 3 assays. And these curves are representing different tissues and sources of these tissues. So we have kidney and lung. And the CQ values we generated from the software were used for our statistical analysis. So first, it's one thing to look at the overlay of the curves, but we want to try to visualize that in another way. So we created a bar chart, and we can see that we're on average maintaining those CQ values regardless of singleplex, duplex, or triplex. And this is shown for the 3 different assays, the MUC1, GAPDH, and the TBP. So looking at our gene expression results, our 3 goals were to compare between tissues, wanted to compare singleplex format versus a multiplex format, and also run-to-run comparisons. We used an ANOVA-based approach with a type 3 test of fixed effects, with our effects being the plex, the tissue, and the run. And the only effect that was statistically significant was between tissues, which is what we were expecting. But we did not see a significant difference in our results between singleplex or triplex, or duplex for that matter. And we did not see a difference in run 1, 2, or 3. So to break that down further, we have below the first table, we have the tissue comparison, the mean MUC1 expression expressed in relative log quantities, where we can see the differences in expression between kidney, liver, and lung. And since that was the only effect that was significant with our first test, we broke that down, and the table at the bottom of the slide shows that comparison between kidney and liver, kidney and lung, liver and lung. And all 3 of those pairwise comparisons were statistically different from each other. And these results matched what we had seen in the publication where we found these assays. So the 2 graphs on the right are just showing the plexing comparison and the run comparison in mean MUC1 expression. And we can see very little difference, and already showed from the first ANOVA table that we did not have a statistical difference between those. So I'd like to summarize the second portion of the talk. I hope this data has shown you that we are able to successfully multiplex gene targets using BHQ probes and the IntelliCube instrument. And again, just to reiterate, these are 1.6 microliter reactions where we were able to multiplex 3 targets in a single well. And we retain PCR efficiency, sensitivity, and CQ values, which are important when looking at gene expression. And so we saw comparable gene expression results between singleplex and multiplex. And so today we are highlighting gene. There's other applications for one-step RT-PCR, so this could be used for detection of RNA viruses and things of that nature. But the other point I wanted you to take home was that regardless of the instrumentation or the platform you're using, when you're looking to multiplex, assay optimization is going to be a critical step in that process. So with that, I would like to thank those who contributed to this work. At our Alexandria site, Mike Freeman, Alex Kolb, and Andres Rodriguez. At our Hoddesdon site, Laura Hinton and Rebecca Howard. And I wanted to thank Tata Biocenter, Christina Lind, and Vendula Novostova for their assistance in our statistical analysis of the gene expression data.
And with that, let's move on to our next speaker for today, and that is Dr. Jim Huggett. I'll pass you presentation control right now, Dr. Huggett, and you can begin when ready.
Thank you very much, Andrew. Okay, so good day, everybody. My name is Jim Huggett, and I'm talking to you from the United Kingdom, and I'm a principal scientist at LGC. And I work in the National Measurement Institute role. LGC also has the UK's designated National Measurement Institute for Chemical and Biomeasurement. If we could go to the next slide, please, Andrew. Okay. And so, as Andrew's already mentioned, I'm also a senior lecturer at the University of Surrey, and with the next pop-up, also managing editor for Biomolecular Detection and Quantification, which is a new journal that I will touch on at the end. So yeah, one job is not enough, I need three. So if we can go to the next slide, please, Andrew. Okay, so I'm going to be talking about a number of different instruments, and I just need to put this slide up here to highlight the fact that this is not an endorsement of any of them, and it's particularly good in the digital PCR world in that we have a number of manufacturers who are really at the cutting edge level and a great group of people to work for. So if I could go to the next slide. Okay, so if we think about the use of molecular methods in infectious disease diagnosis, and I'm talking here now both in preclinical research but also in routine application, things that make changes to people's lives. They're used in a number of areas. So, for example, you can be genotyping, and that may be to look at drug resistance to get some epidemiological information, virulence or tropisms of where the pathogen will end up. Using diagnostics, both broad spectrum detection or more targeted, and increasingly point of care. And in prognostic monitoring, where you are monitoring, for example, the viral load of a patient to guide the clinical decisions and how the patient will be monitored. It's arguable that viral load monitoring, principally using qPCR, represents one of the most advanced examples of quantitative application of molecular methods today. However, calibration of this quantification is very, very challenging, and I will touch on that a bit as we move on. Next slide, please. So, as 50% of you have never done digital PCR and 25% of you are planning to, let's touch a little bit on what digital PCR is. If we consider a qPCR reaction with this schematic as a liquid phase reaction with 10 DNA molecules in this example, digital PCR, if we press the pop-up, is simply a limiting dilution of that process. So crucially, you have, in this example, 21 microliter reactions, and the crucial factor is that some of those contain no DNA. And so you're measuring single or small number of DNA molecules. And if we could go for the next pop-up, or the next two. And this is performed using a normal standard real-time PCR chemistry, and that is primarily probes, but can also use double-stranded binding dyes, such as evergreen. Absolute quantification, which I will talk a lot about in a minute, is simply performed by counting the positive or negative reactions as a proportion of the total number of those reactions, or partitions, as we call them. And you can also apply Poisson statistics to account for the fact that as the concentration increases, you will begin to get more than one molecule in an individual partition. And the measurement community, ourselves, and many collaborators across the world, including NIST in the US, are very interested in this technique because we are counting individual molecules. Next slide, please. Now, digital PCR was originally performed using either a 96-well or 384-well plate. So you're doing a limiting dilution of your molecule DNA across a normal plate, which is both expensive, time-consuming, not very high throughput, and also will give you repetitive strain injury if you continue to do it. The original application of digital PCR predates qPCR and was used in a number of areas in the early 90s. But it is the development of the commercial digital PCR platforms over the last just shy of 10 years that has changed everything. And so if we can flick through these, I think it's 3 advances, and one more, thank you. So you have 2 types of partitioning that allows you to do this limiting dilution in a much simpler manner. You have a prefabricated partitioning using either chips or arrays, or you have solid partitions. And these are examples of the Fluidigm Biomark, Thermo Fisher's QuantStudio 3D, and newer Formulatrix, and the Clarity from JN Medsys. And this is probably not exhaustive now. We do try to keep up. And the other commonly used partitioning method is the droplet partitioning, which is typically oil in water emulsions. And many of you will be familiar with both Bio-Rad and rain dance technologies, and now Stilla have just developed their new NICER system. So there's a lot of development of the technologies here supporting the application of digital PCR. If we go to the next slide. And the reason is there are a number of advantages to qPCR. So, for example, by looking at the distribution of a multiplex experiment, you can look at the relationship of DNA. So you could look at haplotyping in eukaryotic DNA measurements, or, for example, and this has been done quite cleverly, you can look at viral infection and lysogeny in bacteria, for example. Digital PCR has superior sensitivity. It is very good at minority genotype detection, in whether measuring the SNPs that Dr. Linz mentioned, or single nucleotide variants, which are the same type of mutation but occurring at the somatic level. It is arguably more sensitive at the low copy molecular detection, not because it is better at detecting single molecules, but because it is clearer when you're measuring low numbers, as opposed to a slight amplification plot. You get a clear-cut positive partition. It is very, very precise. It provides you with very confident quantification because it is the distribution of the DNA molecules that dictates your precision. In qPCR, it is dependent on the efficiency of your PCR reaction, which can obviously vary. Digital PCR, therefore, gives you a superior fold change measurement. So it may be, or has been applied to look at genome instability in cancer, for example. And the last point is there is no need for a calibration curve to quantify. And it is the minority genotype and the calibration curve I'm going to talk about for the remainder of the talk. My group gives an entire day presentation on digital PCR that covers all of these areas, and this is given a number of times. And so if anybody's interested in this, we can perhaps look at that either by contacting myself or the organizers of this webinar. If we could go to the next slide, please. And there's also a manuscript which is associated with this talk, which will be distributed, published in Clinical Chemistry, which also talks about digital PCR. So digital PCR for minority targeting. So Dr. Linz rather handily gave a great example of how you can genotype using quantitative PCR. Where digital PCR may give you an edge is where your mutation is not very common compared to, for example, your wild type. And so this has been used in cancer genotype measurements, particularly looking at cell-free nucleic acid, where the predominant thing you're measuring is the wild type sequence, or cell-free nucleic acid detection in fetal genotyping. And I'll show you examples of it being used in mutation screening. So in this example here, you have 9 wild type DNA molecules and 1 red mutant. Now qPCR, if you go straight into it using normal qPCR methods, has a sensitivity of approximately 5%, meaning when your mutant goes below 5%, the probe, the mutant probe, will also pick up the wild type at a much reduced efficiency, but that reduced efficiency predominates because there's more wild type sequence. Now, the digital PCR, as you can see for this example, gives the mutant sequence its own partition. And so if you go to the next slide, this can go lower, or to the next slide. With most of with the emulsion instruments can go below this, and certainly with the rain dance instrument has potential to even further low. So you can see, just by diluting, we've done nothing special to the reaction chemistry. We're simply diluting that out so that the mutation gets its own individual reaction, or at least if there is a mixture, the wild type to mutant ratio is in favor of the mutant now. So if we go to the next slide. This is a paper published earlier this year in JClin Micro, and what we're doing here is looking at the importance of digital PCR, the performance of digital PCR, to measure rare or measure mutations that confer resistance to Tamiflu, the drug used in influenza and was stockpiled in the UK during the outbreak in, I believe, 2009. And if it's stockpiled, it's being used. So our collaborators at University College London Hospital had a qPCR assay to monitor the resistance development in the influenza virus. We go to the next slide, please. You'll remember I mentioned that the qPCR is able to measure approximately 5% without any adaptations. And here we have, if you could press the next two forward sets, please, Andrew, an example where we have 1% mutant in a wild type background. And you can see that we're able to pick up the 1% quite easily and distinguish it. So this is just, in front of us an example of how well the digital PCR is able to highlight and detect the mutant sequence. Now, if we go to the next slide, an example from this paper shows how this was used clinically. Now, we did not have a fantastic, huge patient group here, and I would very much like to continue this work within a much larger study. But in this example, you have a patient with influenza over 30 days. And you can see on the left in the blue dotted line, you have qPCR measuring the mutant sequence. And on the right, in the green dotted line, you have qPCR measuring the wild type sequence. And you can see that at the beginning of the measurement, the patient has got wild type influenza, which increases over time, and from the panel on the right. However, the panel on the left, no mutant sequence is detected. However, we do have an increase in the mutation sequence after about 30 days, which continues to increase in the measured period. Now, from this, you might conclude, therefore, that the mutation and the wild type load are increasing together. And whatever you conclude clinically, it would be stretching for me to conclude. However, there is clearly a dynamic being measured here that suggests there is an increase in both mutant genotypes. So we could go to the next pop-up. What this demonstrates here, using digital PCR, is that actually, no, the wild type sequence on the right-hand side is not changing in its abundance. It is on the left-hand panel, in the blue, you can see it's the mutant sequence that is increasing. And what we believe is happening here is that the wild type qPCR on the right-hand side in green is detecting the predominant mutant sequence. And so you can see immediately that the digital PCR is able to demonstrate that the mutant's changing, but the wild type is consistent. And so you're getting a different dynamic from this improved sensitivity. So if we could go to the next slide, please, Andrew. But this is, let's face it, an example of a specialist measurement. And one of the things you'll see in a lot of the higher impact factor papers is how digital PCR is able to do this thing. If you go to the next, I think it's 4 or 3. That's right. Sorry, and if you go back to the earlier slide, thank you. So yes, digital PCR had precise quantification, and it is more precise than qPCR. It can measure this minority detection. There are a number of things it can do that allow what we can do with qPCR to be improved on. But the reality is qPCR is able to do the vast majority of measurements at a much reduced cost and a much higher. So surely, therefore, digital PCR is only useful for specialist applications. Well, we can get to the next slide. First of all, qPCR is an easy technique to do. It's fairly straightforward to get a grad student in and they can start amplifying DNA and getting an amplification plot, and it works very, very well. However, if we go to the next pop-up, and the next one, please, Andrew. Output quantification, whether you call this quantification cycle, cycle threshold CT, or crossing point, is a totally arbitrary unit, and it's dependent on a huge number of things. And so consequently, if we pop up to the next one, experimental standardization is a real challenge. It is really, really challenging. So if we go to the next slide, I'll talk about why that is. So if you flick through these, Andrew, sorry, there's a number of pop-ups. Essentially, this is a qPCR plot. If we go to the next one, you can see here we have two different applications, and the blue line on the left, therefore, you conclude has more DNA in it than the red line. And you could divide the CQ of one over the other or subtract the one over the other and take the efficiency of the PCR or assume it's 100% efficiency to get a magnitude. This is commonly done to measure fold differences, and that's what's being measured. However, you're measuring the magnitude of the fold difference and not the magnitude of the actual quantity. And so you're throwing away a lot of information. Some of you will immediately go, well, hang on, you can run a standard curve. And if we could go to the next pop-up, which of course you can do, and that allows you to give the magnitude to that blue and red line. And if you go to the next three pop-ups, please, Andrew. And the next, thank you. And so you see here is a calibration curve, not like the one we just saw. And so we can conclude from our unknown the log concentration off the calibration curve. However, this is a calibration curve over a vast dynamic range. So subtle changes in that, pipetting, or it can actually influence the quantitative value. Furthermore, for you and me to get the same quantification, we have to make the same calibration curve. Because immediately you can see there are potential issues of using the same calibration curve, having the same, performing the same calibration curve, which can add noise to this method. So we go to the next slide. The other thing with qPCR, if you press a pop-up, is this is kind of what we assume is happening. And we assume it's happening to our clinical sample, and we assume it's happening exactly the same as our calibration curve. And the calibration curve, as we would call, is commutable. And so if we go to the next two pop-ups, this could be happening. There's a whole variety of things that could be happening to take us to the point where we can actually measure the fluorescence. And add to that with the next two pop-ups, different instruments give you different thresholds and different background noises. So you can immediately see that the qPCR quantification is very, very precise, certainly, and it's great in the individual laboratory. But to get each other to get similar results with quantitative methods is challenging. We go to the next slide. The example here, if we flick to two more pop-ups. This is a study out of the NIBSC in Northern London, and they prepare the WHO reference materials. And they basically handed out these materials to a number of clinical laboratories that are measuring viruses and asked them how much is in there. And if you put the next pop-up, the answer is, depending on the virus, the differences are potentially huge. And so if you look at in the middle, you're getting a million-fold change between how much virus is thought to be present. And this is an example of how this is being used every day in routine testing, and that will influence, patient results potentially. We go to the next slide. The reason I think digital PCR is so revolutionary in this point of view is it simply counts the molecules. If you put the next pop-up, please, Andrew. You have 10 molecules. There are 10 molecules there. There's no need for a calibration curve. And this is a subtle potential benefit for digital PCR that I really think is one of its main strengths. If we can go to the next slide, please. And here's an example of some work we did on Mycobacterium tuberculosis, and this manuscript is available in Analytical Chemistry from last year. We can move to the next slide, please. And what we're looking at is simply dynamic standard curves, essentially, by digital PCR. There's no calibration here, and you can see that it is linear and it is precise. If you can go to the next slide, please. And it is, if with the next pop-up, Andrew, it is this strength of digital PCR that is completely unique, and it's this independence of a standard curve that I think is a major, major benefit. And I'll show you why. Next slide, please, Andrew. And so here's another publication that comes out last year on looking at Mycobacterium tuberculosis. And so if we move to the next slide. We have a model system at LGC where we look at the different levels to evaluate the different potential steps of a particular protocol. And so you could look at the gene on its own, you could look at the gene in its genome, or you can look at the gene in its cell. And you go to the next slide. This can be varied to look at different stages of the extraction process and also the individual PCR in its own right. So if we go to the next slide, what we've done here is send around, when it comes, here we go, we've sent around some DNA from Mycobacterium tuberculosis to 4 different laboratories applying different instruments. And so if we could just flick forward, I think it's 3, that's right, you can see that the labs are getting pretty much almost the same result. And if you flick up the next pop-up, if you add all the results together and just do a coefficient of variation, which I appreciate is not necessarily statistically rigorous, it's a coefficient of variation of about 27%, which is incredible. There's no calibration here. You cannot do this using any other method. Go to the next slide, please. Some of you, however, will be standing up on, at the moment, you're going, but hang on, Jim. That's measuring nucleic acid. Nobody really just measures nucleic acid because, as this schematic will show, if you could flick through it, Andrew, to the last, up on the right-hand side, one more, thank you. You have to get your sample. Oh, sorry, if you have to get to your sample, you have to get your nucleic acid out, and that requires an awful lot of steps, which can all contribute error. So this is absolutely right. And yes, we know, if you look at that paper, digital PCR can only measure the DNA or RNA you get to it. And so that is certainly an issue. But if you work that up, it can work very, very well. And in the next slide, I'll show you the same comparison, but this time we're using bacteria, whole bacteria. And you can see we're getting a great comparison again. This is incorporating the extraction. And if you flick the last one up, you can see we're getting a great coefficient of variation again. So again, this is a simple run. We've not done anything clever here. We've simply told the labs to measure 3 different laboratories, and you're also seeing instruments as well. So, this is running through this. There's a huge amount of information here, no calibration. And I really come back to the point that I have to pinch myself that it's actually not possible to do this with any other method. Now, okay, this is a study we've done. It's an infectious disease research study funded by the European Union. And we all are a bit obsessed about this, and it is TB, and we're working with the same assays, using the same extraction methods. So you could potentially say, well, this is an ideal situation. So if we move to the next slide, and it's the close of my talk, here is an example where those 3 labs who did the last bit of tuberculosis work took part in a real external quality assessment study looking at human cytomegalovirus. Now, the virologists, as I pointed out, are arguably apply the most advanced application of quantitative PCR and quantification that is used to make real everyday decisions. And to do this, they need an awful lot of support to ensure that their methods are reproducible and the quantification is comparable, so that HIV measurements in one lab are comparable to HIV measurements to another lab. And I reiterate, this is not trivial. And so here's an example of an EQA study in using cytomegalovirus where essentially you're having a variety of dilutions of the cytomegalovirus which are prepared and sent around to different laboratories. So if we go to the next slide, please. And the next one, sorry. So what you have here are the median values of this comparison. And then in the red lines, you've got plus minus 0.8 logs. The reason you have a squiggly in the line at about 10 to the 4 is that the data set at the higher concentration are many, many more laboratories. And so the next slide shows 11 laboratories who took part in everything using qPCR. Now these are laboratories that perform qPCR clinically on a daily basis, and they calibrate this to the WHO reference material. These are the top-end laboratories, and they are measuring, and this is actually a very, very strong result, where you're getting a dispersion of about half a log between the lowest and the highest value. So these are top-end laboratories. These are using different extraction methods, different PCRs maybe, but they know what they're doing, and they're calibrating this to the WHO reference material. If you go to the next slide, we now have the 3 digital PCR labs. Now, the 3 digital PCR labs are national measurement institutes. They are, how I put it, fairly obsessed with getting the measurement right, but they do not do this on a routine basis. And yes, they are using the same extraction methods, but the point here, and the real big take-home message here, is there is no calibration. Think what we could do if we also calibrate this, and you wouldn't need a calibration curve to do this. This is absolutely, I mean, I cannot, hear my excitement. This is an incredible result. Next slide, please. So qPCR is an analog relative output. Digital PCR is a binary digital output. And if we go to the next slide, or next pop-up, this simply makes it easier for you and me to get a reproducible result. I can produce a piece of DNA, send it to you by digital PCR, and you will measure it, and we will get comparable. We have the potential to get comparable results. This is much, much more challenging with qPCR. If we go to the next slide, as the final main slide, it's a prediction of where digital PCR could be of use both in the short term and the long term. And if we think of our sample, we want to quantify how much DNA or RNA is present, and this could be a virus, for example. At the moment, we have to think about the next pop-up. We would usually do qPCR, and this is, if we think about preclinical research, which does not consider a lot of these reproducibility issues as a routine basis, often you'll do, and so next slide, Andrew, you have to consider a calibrator. And that may be total mass, or it could be, in the next pop-up, fluorescence, which needs a calibrator too. So that would be using the qubit, or other fluorimeter would need to be also calibrated, and usually that is to 8260. So a lot of the calibration we do with nucleic acids is basically to spectrophotometry. Even your qubit is probably to spectrophotometry. There's a circular issue with a lot of the relative methods that they have to be given an absolute value. In routine clinical viral load testing, you have the international unit, which captures the fact that you're doing the extraction as well. But that is a consensus value, which the laboratories have agreed on. What could I do? Well, if we've got the next slide, and there's a calibration issue, so if we go to the next slide, digital PCR could provide an alternative method to calibrate qPCR at the moment. So in your labs that you have high-throughput instruments, you're running phenomenal amounts of qPCR, the digital PCR could be used to support that by value assigning the materials you're using as your calibrators. A major potential role, because it's measuring the same analyte as the qPCR. Ultimately, however, if we get to the next slide, I would predict that digital PCR could begin to be used in routine analysis. The instruments I showed you at the beginning are currently fairly complex. But with the drive and the march of development of technology that happens everywhere and has happened certainly with qPCR, there's no real reason why this could not be simplified. And that digital output would be simpler for a less well-trained individual to measure. It would be much simpler for a point-of-care software to count, is it positive, is it negative, as opposed to has that curve amplified or not. So I predict a lot of potential power for this method. So in my last slide, certainly my last non-acknowledgement slide, I would just like to point out, however, that there is an issue of digital PCR that we need to consider, however, which is not to run and use it as the kind of fantastic method that will save all problems at this point. It still has issues we need to consider. Will digital PCR require calibration? Probably. It might be a lot simpler than qPCR, but I would not, coming from a measurement institute, suggest that this measurement is necessarily calibration-free, certainly not in a routine end-user laboratory. And so as we apply digital PCR and publish on digital PCR, we need to be mindful of what we are doing. And one of the big problems with qPCR that has meant that while there are tens of thousands of publications purporting to perform qPCR to quantify something, where that is used in routine practice, whether that's clinical practice or whether it's looking at foods analysis, is quite rare. considering if you were to do a ratio of the published papers and the number of routinely applied applications of qPCR that mean a difference to you, it is quite rare. And I think one of the reasons for this, as well as the standardization problem, is the fact that we publish without the details. And for this reason, the MICA guidelines were published in 2009 to support that. And we published the digital PCR guidelines in 2013 to support the development and publication of digital PCR. And if you're going to perform digital PCR, I would implore you to look at this manuscript. It's freely available from Clinical Chemistry, and I believe it's available as part of this webinar, because it talks about how to do digital PCR, and it also talks about the things to consider when you are publishing. So that's me pretty much done. I would just like to go to the acknowledgement slide. Thank you very much. And I have a huge number of people to acknowledge. My group and my team and my colleagues are large, and they're all very, very top people. Alison Devonshire and Alexandra Whale are particularly expert in the digital PCR area, and Simon Cowan is a fantastic statistician I'm fortunate to be able to work with. University College London, Professor Tim McHugh and Isabella Honeyborn, very helpful with the tuberculosis work, and Jeremy Garth and Catherine Harris and the bacterial work we do. Heinz Zeikart and Hans-Peter Gruner are fantastic virologists I get to work with on the cytomegalovirus work, and Versel and Pablo Mendoza in support of the tuberculosis work. And I am very fortunate to be able to work with the vast majority of the digital PCR manufacturers, and they're a great bunch with this particular work. Though Bio-Rad have helped quite a bit with the influenza work I showed you, so I'd like to thank them. And then there's the JRC and NIB who also do the digital PCR work with us as part of that calibration. I'd also like to thank the funders, European Metrology Research Programme and the National Measurement Institute, and we have a wide collaborative network in Europe, and despite recent occurrence in the UK, I'm certainly European. So if we just go to the final slide, please, Andrew. Which comes back to the journal I was telling you about, which is Biomolecular Detection and Quantification, which is very much linked to championing improvements in molecular measurement, whether this is digital PCR, qPCR, next generation sequencing, etc. And we are very much welcoming publications and manuscripts, so please consider us. And I thank you very much for your attention, and I apologize for the problem at the beginning.
Well, fantastic. Thank you very much for that, Dr. Huggett. Also, thank you to Dr. Linz for his presentation. Both of those very insightful and exciting. At this point, I'd like to invite our audience to continue sending in their questions and comments right now using that questions window. For this, of course, is the Q&A portion of the webinar, and we've already got a lot of questions here, so let's get right into it. Our first question here is for Dr. Linz, and that question is regarding the singleplex SNP genotyping slide that you presented earlier on. Why were signals for heterozygotes so low?
Yeah, so that can be explained. With a heterozygote, you're actually getting competition between the probes, so your competition for your PCR agents when you're amplifying the two different targets. So overall, you're going to get a slightly lower signal when you have a heterozygote sample.
Okay, thank you for that. Moving on to another question, also for you, Dr. Linz. How do you evaluate primer dimers and interaction when you design your multiplex assays?
So there are a few different software programs available. Some are free, some are not free. Just to give you an example, one of them is from Premier Biosoft and NetPrimer. So basically there's programs where you can input your primers, and it'll give you an analysis on the interaction between those primers. So if you want, I could provide you with some more detail. I'll have to go back and check on that, but if they're interested, we can provide some of those programs or show them where they are.
Excellent. All right. Well, I'm sure they'd appreciate that. Moving on to another question here. Do you have an explanation for the fanning effect of too much primer?
Yeah, we think it's just interaction between the assays. So when we had too much primer from the other assays, that was interacting with the TVP assay. So it's sort of inhibiting that that particular assay causes those curves to fan out. So reducing the primer concentration reduces that non-specific interaction that's happening and causes the curves to perform as were intended.
All right, thank you for that as well. Moving on to another question, also for you, Dr. Linz. This question, this audience member writes, looks good for triplex. Have you tried quintuplex or septuplex? Does one get diminishing returns as complexity is increased? Or is there a plateau of performance with multiplex? Pretty solid. So your thoughts on that?
At this time, we haven't evaluated past triplex. There definitely probably will be a point of diminishing returns. Again, that might go back to how good that master mix is. Do we have a sufficient supply of all the necessary components in the mix? I imagine, though, that we could definitely add additional targets, but I think each time you're adding more targets, it's going to be a little more challenging to optimize. If an individual is willing to put in the time to do so, depending on what their application is, it's probably possible at this point. Again, our detector only has 5 channels, so the most we could do without assuming we don't use a reference gene would be 5 targets on our current system.
All right, good to know. Thank you for that. Also, another question for you, Dr. Linz. We had lots come in during your presentation. Can you test larger volumes?
At this time, our system is set up for just the one volume of 1.6 microliters. Our array tape manufacturing is capable of producing array tape with larger volumes, but the IntelliCube is designed to support our current 1.6 microliter reaction at this time. Okay, excellent.
Moving on to another question, let's perhaps move to some questions for Dr. Huggett. I believe. How do you deal with concatamers?
Okay, thank you. So I'm guessing this is linked to the fact that digital PCR cannot measure multiples on the same molecule because they'll be in the same partition, which is a disadvantage. If you're looking at concatemerization, for example, in HER2 amplification, which is measured as a potential biomarker in breast cancer, then you would need to either fragment, either using physical fragmentation or restriction digestion. And that is certainly a potential, adds a bit of complexity to it. We also did some work which we published, I believe, in PLOS ONE to demonstrate you could also do a preamplification. However, doing the preamplification would reduce the precision you demonstrated that you'd need for improving that measurement. So restriction digestion or physical fragmentation is essential if you want to accurately quantify molecules that are amplified in cis. That's certainly the case. Although that is essentially when you're looking at 2 different molecules that are together in cis for haplotypes or integration, then that's an advantage. But that's a fair point.
Interesting. Okay, thank you for that. Moving to another question here for you, Dr. Hoggett. Do you see evidence of Poisson distribution of PCR inhibitors in the digital PCR?
Now, if I understand the question correctly, are we getting Poisson distribution of the inhibitors? I do not think so. We don't see any effect. Where we've looked at inhibition, what is quite frightening for me from the point of view, well, any measurement, is in a PCR, if you inhibit the reaction and kill the reaction, it's going to affect the reaction. It's not going to work. In qPCR, if you have a mild inhibition, you can get a right shift of your amplification, and so you get a higher CT or CQ, and you may maintain the precision. So unless you're running some kind of control, then that could be a real problem. The general consensus is that digital PCR is less susceptible to inhibitors than qPCR, and our work demonstrates that with many of the inhibitors we've looked at, that is certainly the case. However, we've also demonstrated that digital PCR can be susceptible to inhibitors that qPCR is not, and probably the thing that we most be worried about is you can get the equivalent of a right shift with digital PCR. So you can get a reduced count, which has a very good distribution, where you're getting molecular dropout of a large number of the molecules. So it looks, to all intents and purposes, like a perfectly good reaction, but it isn't. You're measuring a small proportion of the DNA molecules because of the matrix effect. And this is published as well. And so that is, I mean, that can be available. If anybody's interested, I can send that paper or send the reference to that paper.
Interesting. Okay. Well, thank you for that as well. Moving towards another question here for Dr. Linz. You mentioned optimizing primer concentrations for multiplexing. Do you need to do this optimization every time you have new mixture of targets?
Yes, likely you're going to have to if you're running a new set of assays. We can get a pretty good indication of how primers and probes are going to react using the software, but a lot of times it does take some trial to see how those assays are going to behave together. So usually if the user is interested in it, if you're looking at something you're going to run on a routine basis, that's when multiplexing may be worthwhile. If it's something you're going to be running just here and there, sometimes you have to consider whether that's worth your time to do that optimization, depending on what you're looking to do.
All right, excellent. Thank you for that. Moving to another question here for Dr. Huggett. So digital PCR could support qPCR in a clinical laboratory by validated reference materials, but will it ever be used directly on patient samples? Your thoughts on that question?
That's a fair one. It ultimately depends really on how the instruments are developed. So I guess it's how we as a community apply it and use it, and that therefore gives the vendors the incentive to further develop the instruments. They've done a fantastic job thus far, allowing us to perform digital PCR and just to demonstrate how well it works. And I'm not selling this instrument. I really believe in this technology. I think it has a lot to add. And as I mentioned at the end of the presentation, it lends itself to the output rather, not the current format, would lend itself to a point of care situation where you do not need such complex algorithms to measure it, or an individual who's perhaps less well trained. Are these positives or are these negatives? It's easier for the instrument to call the results. Certainly, from a quantitative point of view. So yes, I think it could well be used. I think it has a lot of potential. It just depends on how we use it and whether there's incentive for the vendors to further develop it. But I hope they do.
Okay, excellent. Thank you for that. Maybe time for one final question here. Will the next generation sequencing not make digital PCR obsolete? Yeah,
this is okay. My response to that, and in the clinical chemistry manuscript, the review we published on the application of digital PCR is next generation sequencing will ultimately make PCR obsolete. It's a question of when. Now, if we look at PCR and the application of PCR clinically in a routine manner, it has not really been applied as well as perhaps it should be. We know that PCR, for example, can diagnose tuberculosis. It has been diagnosed it's been demonstrated to diagnose tuberculosis potentially maybe 2 or 3 years after PCR was initially published. There's a paper on it. But it's only really the last 5 or so years that we've actually got together, and Assay primarily have got together to be able to diagnose tuberculosis in a routine manner in a near-patient setting. So it is possible. PCR is quite simple, so why has it taken 30 years for that to happen? Next-generation sequencing is not that simple. So while I believe NGS will replace PCR, and PCR, let's face it, is a method we use where we have to generate 10 billion molecules, so our optics, which are pretty bad, are able to measure anything. The question is when, and so I still think PCR has an awful lot to add, especially as many of the next generation sequencing methods are currently depending on PCR. This reproducibility of digital PCR, I may have a silent view on it, or talking to the wind, but it is really, I see a lot of potential for that. I really do. In physics, in chemistry, the need for reproducibility, the need for you to get the same result as me is central. We don't have this in biology yet, and it's some of the work that the National Measurement Institutes are doing. But if we want to translate our research, we want to have impact on patients, we need to worry about reproducibility because we need to be measuring comparable results so that we're getting the same setup. So I do not think next generation sequencing will make digital PCR obsolete. I may be wrong. Perhaps it'll be a good thing if I am wrong because it'll allow us to do a lot more and help a lot more people. But in the short run, I still think there's a lot of potential for this method.
All right. Well, excellent. Thank you very much for that, Dr. Huggett. Also, thank you to Dr. Linz for all those answers. We have reached the end of the question and answer portion of this webinar. But if you do have any further questions, you can direct them to the email address showing on your screen right now. That's marketing@biosearchtech.com. And if we weren't able to attend to your questions today, the team at LGC Biosearch Technologies will try to follow up with you after the webinar.